So this is the key structure of a plant cell. So I'm gonna move this out the way for a second, and then kind of highlight our prokaryotic cells. So with a bacterial cell, what you essentially have is the existence of these protein molecules. Let me get that out of the way. Where you have the phospholipid bilayer. So you have you have a membrane surrounding the cytoplasm, which provides a foundation for the structure of a bacterial bacterial cell wall. Okay, and then above the cytoplasmic membrane, which again is just phospholipids that surround the cytoplasm or the cell, you have a structure called peptidoglycan. And I know I've talked about this just a little bit, but here we're really going to dive into the details of peptidoglycan. With peptidoglycan, you have these small little peptides that hold together glycan chains. So like I showed you in that plant cell image, you have basically these sugar molecules of glycan that are held together by peptide bonds. And this chain linkage will provide support and protection to this cell to keep the cell from succumbing to osmotic pressure. In this particular structure, remember I've said before that we have gram-negative bacteria and gram-positive bacteria. Gram-positive bacteria will have 40 layers or more of peptidoglycan. And here we only see one layer of peptidoglycan. So this says to me, when I see this one layer, this is called a gram-negative bacterial cell because it consists of one very thin layer of peptidoglycan. And some gram-negative bacteria don't have any peptidoglycan, so it kind of depends on the species. But gram-positive bacteria, if I were showing you an image of that, you could expect to see 40 layers of peptidoglycan or more in the bacteria. And that's important because it's this peptidoglycan structure that we use as a structure that's targeted for destruction via an antibiotic. So if we wanted to kill a bacteria that had very thick layers of peptidoglycan, we could easily attack it with penicillin, and that penicillin would disrupt the peptide bonds that exist in this peptidoglycan chain, and that would allow this whole bacteria cell to break down. So that we'll save that for another lecture. But just know that peptidoglycan, if you see that inside of a cell, that you're looking at a bacterial cell. Okay, and this particular diagram here, this is an example diagram from a gram-negative bacterial cell, and I know that because it has one thin layer of peptidoglycan, and on top of that thin layer of peptidoglycan, we also have a structure that's referred to as an outer membrane, and only gram-negative bacteria will have an outer membrane. Gram-positive bacteria do not. So in the outer membrane, this consists of specialized biomolecules known as lipid A, which is a lipid and just referred to as lipid A. And above lipid A, it's affixed to a polysaccharide that's known as O polysaccharide. Okay, so this lipid A affixed to an O polysaccharide makes up a structure known as lipid polysaccharide, which is also referred to as LPS. So your outer membrane is essentially a phospholipid bilayer, which is comprised of LPS. And LPS is important because it actually has toxic properties to a cell. So not in the sense if a bacterial cell landed on your skin and it was gram-negative, it would cause LPS to cause you to have an allergic reaction. But what happens with bacteria that are gram-negative and have LPS is once this bacteria is destroyed, all this LPS can be released into the host organism. And as it's being released, it will generate toxic properties and harm the host that it's attacking. So you essentially have a dead bacterial cell that can release LPS and still do harm to the host cell, which is why we've entitled this specific slide here that you can see as kind of zombie bacterial cells, because they do their worst damage to a host when they're dead. So again, you can have an antibiotic, or you can have a structure inside of a cell that will, like that peroxisome that we talked about being in the eukaryotic cell, that will degrade this outer membrane of this gram-negative bacteria, and it will release these toxic O-lipid A and O-polysaccharide components. And when they are released into the host system, then the host essentially can be attacked, and it's being attacked from a dead bacterial cell. This is something I think is kind of interesting regarding an attack mechanism. But that's not the focus of this particular unit. We're just focusing on structural components. So in terms of gram-negative bacteria, make sure you know that you have a very thin layer of peptidoglycan, and you also have the existence of an outer membrane that is made up of LPS, which consists of lipid A and O-polysaccharide. Gram-positive bacteria, we do not have outer membrane. We just simply have 40 or more layers of peptidoglycan surrounding the cell. So that's the cell wall of gram-negative bacteria. And we talked about the cell wall of the gram-positive bacteria. There's not much to say about that other than it has no outer membrane and thick layers of peptidoglycan. But the cell walls in organisms, just to kind of recap that idea, remember, cell walls are not found in all cells. You have cell walls present in plants, algae, fungi, and bacteria. And there are some bacteria that don't have a cell wall, like mycoplasma bacteria, but we'll talk about them later. But the majority of most bacteria are all going to have a cell wall. And then in animal cells and in protozoans, and then the mycoplasma bacteria that I just spoke of, these do not have cell walls. Okay. Another structure that can also be found on eukaryotic cells and prokaryotic cells is that of flagella as well as cilia. These structures can be used for motile purposes. A flagella in particular, I'm sure you've seen this if you think of like a spermatocyte, which is a sperm cell. It contains a whip-like flagella that helps to prepare the cell through fluid. There's a lot of protists that have flagella, and there's some bacteria that have flagella, like the organism called Salmonella, or the genre that makes up organisms residing in the Salmonella genre. So the flagella is simply used for locomotion purposes. And different species of bacteria or different protists, they can have more than one flagella. There are some organisms that have two flagella. There are some that have many flagella. There are some that have 3 flagella. There are special names and terms that we'll go over in just a minute regarding, how many flagella a specific cell has. But the key caveat here is that flagella are used for motility purposes. And some cells are also able to move the structures known as cilia. I'm sure you've heard cilia before, probably in your A&P classes. Cilia are hair-like structures that exist or surround a specific cell. They can be very short and very thin, and they often are going to cover the entire surrounding membrane of a cell. So this also again enables a cell to be motile, so that way it can move. We have cilia or microvilla that line our cells of our trachea, which are called pseudoepithelium cells, but they have these hair-like structures on them, not for motility purposes, but for capturing elements inside of our trachea that we don't want to go down into our bronchial tubes. So you think of them as kind of a filtration device. But in microorganisms, we typically see cilia on protists. We can see them on things like paramecium, which are very interesting organisms if you've never seen these underneath the microscope. And the cilia are used for motility purposes for most microorganisms. Here's an enhanced microscopic view of some cilia. The cilia consist of these specialized structures that are called microtubules. They're just very small microtubes. That's pretty much all they are, and they're jumbled together, and they look like hair. So in some cells, like those that lining our trachea, they can be used for filtration for a more complex host, but for primitive hosts that consist of one cell, which are unicellular, such as protists, they're primarily used for motility purposes. Okay. Let's talk a little bit about, the prokaryotic cell structure, because we've spent a lot of time talking about eukaryotic cells, but we really want to get to the heart and meat of our core requirement, which is that of prokaryotic organisms. Prokaryotic cells, remember I talked about when we talked about microscopy, are incredibly tiny. They are going to be 10 times smaller than a eukaryotic cell. So if you compare one single skin cell to one single bacterial cell, you're looking at a eukaryotic skin cell being 10 times bigger than a prokaryotic cell. Prokaryotic cells, like I said, they're very low maintenance and very simple. They consist of nucleoid region, and they consist of ribosomes and cytoplasm. The eukaryotic cells, we spent a lot of time talking about their complex machinery that enabled them to require a lot more energy to grow and develop. Prokaryotic cells also undergo an interesting kind of reproductive process that's called binary fission, which is essentially one cell splitting itself into two cells. There's essentially two groups of the prokaryotes, which I've talked about. We have the bacteria group, and then we have the archaea group. And all the members of the archaea and bacteria group are considered to be prokaryotes, which means they do not contain a true nucleus. The cytoplasm that's found inside of prokaryotic cells lack internal structures. This is supposed to be prokaryotic. Sorry, I'm going to change this real quick. So we don't have any structures inside of the cell, inside of the cytoplasm of a prokaryotic cell that's going to contain any organelles to them. You'll see ribosomes, but those ribosomes aren't going to be surrounded by internal membranes. The cytoplasm in the prokaryotic cells is going to be surrounded by a membrane. That's also going to be referred to as a cell wall. And then in some cases, in some bacteria, there's an additional layer that surrounds the cell wall that also is used for protection purposes, which is called a capsule or a slime layer. And the capsule prevents cells, and such as in a human body, that can phagocytize a bacterial cell from eating it. So when a bacteria, to protect itself, a bacteria can invest energy in creating a capsule or a slime layer. And so when a macrophage or a white blood cell comes into contact with a bacteria that contains a capsule, it can't digest it because the capsule is, it's a very strong, sticky complex that's hard to penetrate through. And so here's the simple diagram of a prokaryotic cell. So it looks a little bit more complex now in this particular figure, but really isn't all that much complex than the eukaryotic cells that we've talked about. There are essentially 3 layers that are going to make up the outer covering of a prokaryotic cell. And like I said, there's some layers that aren't in existence for all bacterial cells. All bacterial cells will have a cell membrane and they will have a cell wall, but not all bacterial cells will have a capsule to them. So there's actually a small number of organisms in the bacterial world that contain a capsule. But on this diagram it's present, so we're going to talk about it. As I said, the capsule is used to prevent the digestion of this bacterial organism from a host organism. So the capsule surrounds the cell wall, which, remember, is inclusive of that peptidoglycan structure, or LPS, that might exist in a gram-negative bacteria or outer membrane. There's the cell membrane that surrounds the cytoplasm of the cell. You have all this chromosome, nucleoid, or nuclear genetic material that exists inside the cell. And this chromosome looks like it's really big and really long, but really it's just a spherical chromosome that's been coiled. You also have plasmids in here, which are also known as circular DNA, and then you have flagella that enable motility. But not all bacteria have flagella.